Okay. Hello, everyone. Good morning. My name is Ramesh Goel. I am a professor of environmental engineering at the University of Utah, Salt Lake City. Um, I am going to present on something we have been working for the past 7 to 8 years. Initially, we did a lot of work related to Utah Lake sediments, and then more recently, over the last 4 or 5 years, we have been working on harmful algal blooms in general, their relationship with different environmental factors, and then the ecogenomics. So just to start with, importance of freshwater lakes, only 3% of the water on Earth is freshwater, and of this 3%, only 0.3% approximately is available to us. So all we see in the form of Great Lakes and other freshwater rivers on Earth is just 0.3%. So you can appreciate the importance of preserving, conserving, and protecting our fresh surface water. Excessive phytoplankton growth, what is this? So the question is why Utah Lake turns green, and this is because of excessive growth. This is important. Almost all surface water bodies, to some extent, they have, phytoplankton growth. But when they're excessive, the water body turns green. And this happens because of these unwanted contaminants, what we call them nutrients. And the sources of nutrient could be non-point sources, which are coming from agricultural runoff, tributaries. For example, in Utah Lake, there are several tributaries coming and contributing their flows, to name few, like Spanish Fork River and Provo River, and many more. Atmospheric deposition, precipitation, this is a big question for Utah Lake, and then anthropogenic sources also. And in the form of point sources, municipal wastewater discharges, runoff from construction activities. And the relative contribution, the contribution of each of these sources is important for evaluating the eutrophic status and the pollution status of any water body. So, as I stated, when these algae altogether, cyanobacterial algae, they grow in excess. This excess growth leads to eutrophication, and a part of algae, phytoplankton, we call blue-green algae, is known as cyanobacteria. So excessive eutrophication results in cyanoblooms, algal blooms, and cyanoblooms. These are two different terms. Algal blooms refers to all algae, including cyanobacteria, and then those blooms, when algal blooms are dominated by cyanobacteria, they're known as cyanoblooms. So cyanobac blooms is a global problem. It exists everywhere. As you see in China, of course, there are several hundreds of papers came about eutrophication in Lake Tahoe and other important lakes, Russia, and of course here in West Coast also. And this is a map from USGS, and see everything is seems green except some some areas. And then you see right here we see Utah, and there are like several episodes of algal blooms in Utah Lake, and Great Salt Lake, Farmington Bay, and then other parts of some reservoirs also. So just to give a little background, what are phytoplanktons? They're photoautotrophs. As you see, photo means light, and autotrophs means those who survive on inorganic carbon source. That means carbon dioxide gas. So, and then other important thing is they need several other micronutrients, but on a global scale, these two are very important. P stands for phosphorus and N stands for nitrogen. Phytoplankton could be macroalgae and divided into microalgae, small one, microscopic count, and then in microalgae you have eukaryotes, and then prokaryotes, bacteria, and cyanobacteria belongs to prokaryotes. And further, what we worry about is not only aesthetic nature of water body, but cyanobacteria have huge, tremendous human health implications. And this is because of their toxic nature. When I say toxic, all bacteria, they survive on nitrogen, carbon, and phosphorus, plus other micronutrients, some metals, for example. But they produce these toxins as their secondary metabolites, microcystin, for example, and then Xenotoxins. And these toxins are, but not all cyanobacteria produce these toxins. Some specific strains, group of cyanobacteria, they generate these cyanotoxins. And then the non-toxic strains I stated, and Xenococcus, for example, and then some strains of phenylnogeminone. So, and then other important thing about cyanobacteria, are they could be nitrogen fixers and non-fixers. And that's where the contention and the argument among ecologists is, which nutrient is of limiting, which nutrient is limiting, whether it's nitrogen, which means which one is important, it's nitrogen or phosphorus. And often cases, there are two groups of people. Some say, they are the nitrogen limited, mostly freshwater lakes, sometimes phosphorus, and then many times people have found they're co-limited. That means nitrogen and phosphorus, both are important. But this is most important thing that, many cyanobacteria, they can fix nitrogen, which means bring atmospheric nitrogen and then, use it and then convert it into ammonia. It's called nitrogen fixation. And then non-fixers, of course, they can't. So just a little bit of history, origin of, the cyanobacteria, they existed, even billions of years. It's a say billion year, BYA 3 billion years. So when Earth was pretty hot, CO2, abundant CO2, and as I stated earlier, they consume CO2. So it's believed that they contributed a lot in lowering the temperature of Earth down. They consume CO2 and they generate as a byproduct, because they're autotrophs. Photosynthesis means generating oxygen and taking up CO2. So they produce oxygen. As a matter of fact, then we have oxygen to breathe in the atmosphere. So these are some of the pictures. This one is most often found in Eurolake phenylnogeminone flux. These are, as you see, long strings. They are filamentous in nature, and then some of these, Xenococcus. These are called picocyanobacteria, not even nano pico, very, very small. And these microcystin, these are the culprit which they produce cyanotoxins, microcystin, LR, and different conglomerates. So as I stated, cyanobacteria, the different groups, pico, cyanobacteria, filamentous, just based on morphology, and colonial cyanobacteria. And that's how people, using microscopic technique, they identify and they affiliate their phylogeny, and they say, okay, this water is dominated by phenylnogeminone, which is pretty powerful technique, microscope-based techniques. So, but their structures, combination structure is dynamic. The challenge is they change, over days. For example, Utah Lake, you will see, May end, it comes June. It's pretty clear, little bit greenish, and then suddenly it comes June end or July, and it suddenly turns green. You see algal blooms occurring. So conditions were same. Temperature plays an important role in the domination of different cyanobacterial groups. So again, little bit about cyanotoxins. These are secondary metabolites, and the complex organics, as you see, that very, very complex structure, and then some are freshwater. For example, nazurine, it occurs in brackish water. So if we talk about Great Salt Lake, you will find, nazurine, if those bacteria exist, of course. I'm not saying nazurine always exists in Great Salt Lake, but this is brackish water, and then other are freshwater primarily. So different structures, and so cyanotoxins, as I stated, these different bacteria contribute. Cyanobacteria contribute to cyanotoxins, anabena, and then some are hepatotoxins, neurotoxins, and there are dermatotoxins also causing irritation to skin. And that's why cyanoblooms are not only important, or the understanding is important from the point of view of environmental sustainability, but from human health point of view also. So the question is why Utah Lake is green, and this is, cyanobacteria cell count, courtesy by Utah Division of Water Quality. And as you see right here, the cell count of cyanobacteria increase. That this is where that big notice was issued by State Health Department, that not to swim, and there were beach closures where there were like cell count exceeding million, 2 million per mil. So not in liter, but one small volume, and that's why this is green. So, and then based on the cell count, again, this is not genomics. This is all everything from Division of Water Quality website. So these data you see, right? I mean, in Provo Bay and some area, Utah Lake State Park, Lincoln Beach, as high as even 10 million cells were observed, and these are some of the sites which DWQ sampled. And then based on microscopic identification, they found phenizomenone, microcystis, dolisopramum. These are all, these, for example, these two, they are toxin producer, anabena also. So why is Utah Lake green? Challenges: nutrient enter the lake through stormwater discharges, as I stated, common factor. Tributaries such as Provo and Spanish, my apologies for the typo, should be Spanish Fork River, agriculture runoff, point discharges, atmospheric depositions. And so, as I stated, microscopic methods, you take a sample and then you process, and then you add some chemical to preserve, then using this slide, which is labeled with these different metrics. Then you count the number of cells actually, and sometimes people also do area analysis based on their either the average diameter. They can do area analysis also if it's too heavy. And then because the morphology of known, again the word is known, cyanobacterial strains is they're all defined. So when you look under microscope, they look at the chart and say, okay, this resembles most to phenizomenone, so this is phenizomenone, which is pretty powerful technique, and the morphologies are well established. The challenge is when you have number one, known, unknown strain, whose morphology is not defined, then it doesn't catch. And if more than two strains, different strains or different even phylum, they have same morphology, then they go under same division, same category, and one could be toxic and other could be non-toxic. So these are some of the challenges with microscopic techniques. So I summarized here what are the challenges. Smaller cyanobacteria in microscopic technique, this is one of the main challenges, and you will see in Ecogenomics some of the results I'm going to share today. That the data, microscopic data on Utah Lake showed that the lake was dominated by phenizomenone. Of course, it was present, but I will share some of the data. So smaller cyanobacteria, when I say smaller, means picocyanobacteria, escape microscopic count. You generally don't see them because they're so small. So what is Ecogenomics framework, which we have been using in the lab, and then this is widespread and around the globe, people have started using this ecogenomic framework. And eco, as state, ecology, diversity, and genomics means using at the cell's genetic level. As we know, the two most important nucleic acids are RNA, ribonucleic acid, and deoxyribonucleic acid. So DNA, what we call genomic DNA, is used to profile microbial community ecology, and then, or what we call the taxonomy. And then physiology, that means presence doesn't mean something is, they're doing something. They could be flourishing, chewing upon carbon, nitrogen. For example, some cyanotoxins bacteria, the present doesn't mean that it is contributing to cyanotoxins. So to understand whether they are doing this or not, then we do the RNA extraction, what we call messenger RNA, and we use this technique to correlate this taxonomy, ecology, that means their presence, with their function, whether they're functional or not. So this pretty powerful technique. So DNA, is double-stranded. It's connected. Each strand is connected through H bonding, and then each what we call bases, and there are four bases: adenine, thymine, and then guanine and cytosine, and they are the sequence how these are placed, how these bases one after other placed in one strand constitute a DNA sequence. And then the placement, that sequence, is what makes one bacteria different from other, or one human being or one animal different from other, the sequence. And the A always forms pairing with T and G, and vice versa. G always forms pair with C. And then by extracting DNA, eventually the aim is using this technique. And this, I am giving you an example. Let's say one environmental sample initially had 6 bacteria. So we extract DNA, but the DNA is hodgepodge. That means just after extraction, you cannot separate DNA just from blue bacteria, DNA coming from red bacteria. They all are present in one small volume. And then the sequencing is done. The sequencing again provides you the small reads, which are again coming from, some reads are coming from red bacteria, some are coming from green, some are coming from yellow. But again, they are present in all together. They doesn't come out of your sequencing machine one by one. So now you have this hodgepodge, a mixture of these different reads which you don't know. So using computer tools, what we call bioinformatics, Python script and Perl scripts, advanced computing, then we separate these reads. The reads which belong to, say, for example, red bacteria, the reads which belongs to green bacteria, like that. And then those reads are separated, and then because these reads are smaller, the sequencer doesn't give you big reads. That gives you 150 to 300 base pair. But the bacterial genomes are pretty large, like few million base pair. So what we do is, using again bioinformatics, we combine these small reads into bigger reads, and then this is what we call, and those bigger reads are used for taxonomic classification, and this is what we get eventually. And this doesn't give you absolute number, this gives you relative abundance. That means in between samples. So as this diagram shows, the taxonomic composition at this label, yellow belongs to one bacteria, cyanobacteria, then this light blue, and then likewise red belong to this different bacteria. This is what we get from in spatial as well as temporal time scale. And then we can go at resolution also, as we know. We can go from like bacteria, the overall domain, to the phylum, and then all the way up to genus using this technique, and then even species also. So this is pretty powerful technique. And then, as I stated, instead of 6 bacteria, what if the environmental sample, which is the case most of the time, has thousands and trillions of bacteria altogether? And the methodology is similar. We use bioinformatics, separate each, and then combine them using extensive bioinformatics. So present doesn't mean they are active. So as I stated, other molecule is RNA, ribonucleic acid, and then this mRNA, this is called transcription, which is a process where this RNA polymerase synthesize this mRNA. That means the genetic information sitting on the chromosome or genomic DNA is now transcribed, and that transcribed information is carried by this messenger RNA for translation, translation, conversion to protein. Protein is nothing but these particular enzymes, right, which catalyze different reactions. So bacteria, they act upon different contaminants because of these enzymes. Some bacteria, for example, can degrade acetate because they have acetate degrading genes sitting there which are expressed. Some bacteria, they can degrade glucose because they have. So this is a very simplistic example. So RNA, so this expression of mRNA, this is what we do. From any environmental sample, we extract mRNA, which encodes for the expressed gene, not the gene present, but mRNA only tells you what gene is expressed. That means the functionality, the true functionality, and that mRNA is converted into cDNA, and then we sequence it. And by sequencing, we can tell that in this environmental sample, this particular functional gene was expressed, this particular functional gene was expressed. So we can track. This is called transcriptomics. So quickly to Utah Lake sampling, we sampled 4 sites in 2016. And I'm not presenting data from 2013 and 14, where we did extensive sediment sampling and then mineralogy, for example, because one of the things about Utah Lake is much of the nutrient, especially phosphorus, sinks to the bottom of the lake. That particular, I would say, observation needs further experimentation, but this is because of the nature of the lake, because it contains a lot of carbonate and calcium. Everything settles at the bottom. But how much and what extent needs further investigation, of course. But in terms of genomic analysis, we did extensive sampling in 2016, 2017, and these are some sampling location. And then, so high throughput sequencing, as I stated, we extract DNA, and then instead of just focusing on one bacterial group, we sequence all different bacteria present in the water sample, and then do bioinformatic analysis. We do ecological analysis, and then come up with this nice relative abundance graphs. So some of the water quality data, 2006, June and July was the time when the lake was blooming, and this is chlorophyll A. And then again, this is chlorophyll A representing high concentration at some of the sites. You see Provo Bay, which is shallow part of Utah Lake, and then it's pretty high right here. Some sites were blooming in June. Unfortunately, we did not have data in these 2 sampling because of some glitch in our sample collection. But then for other months we did. And then likewise for 2017 also, this is chlorophyll A. And right here you see, in July when the lake was blooming, all sites, mostly all sites were pretty high in chlorophyll A concentration, telling that there is phytoplankton growth. But this chlorophyll A concentration, high concentration, doesn't mean that there are like bunch of cyanobacteria. That only tells that there's a lot of phytoplankton growth. So 2016 data, this is at phylum level. You recall, you can go up to like genus and species level. So this is like phylum, bacterial phylum. So 4 sites: Linden Marina, mouth of Gosen Bay, mouth of Provo Bay, and Saratoga Spring. And this one, light orange right here, this is Proteobacteria. And Proteobacteria is a very common phylum, which you see in any environment, you will find it, because very useful bacteria. Most of them, they belong to Proteobacteria. It's very, very robust phylum known in bacterial domain. And then the important thing is Cyanobacteria, this green one. So again, this is at phylum level, but why this technique is also powerful. This not only tells you about Cyanobacteria, but it also tells you about what other bacteria are present in your environmental sample, in this case, water sample, water column. So apart from other bacteria, let's see, in June, Cyanobacteria is equally abundant, almost 20%. And then right here in July, for Linden Marina, this blue one, bacteria, they are abundant, more abundant than Cyanobacteria. So we'll see that at different sites, the Cyanobacteria was present, and in some location it was present in very small quantity. The small green strip, this is what Cyanobacteria is. And then at the genus level, that means Cyanobacteria, this is all Cyanobacteria now. You see different Cyanobacteria. And then one thing I just want to point out, the interesting thing is, look at this orange, this one is, this green one is Phenizomenon, which was established through microscopic measurements. But look at this Xenococcus. Microscopic measurement did not show any Xenococcus. This is Picocyanobacteria, but all sites. And then it was more abundant than Phenizomenon in month of June and July, even August, then other cyanobacteria. And then you see this yellow stuff also Dolichocrospimum, and then the Planktothrix also, Planktothrix right here. You see late August. And we have seen this trend. And initial month of summer, you have this filamentous bacteria, and the story goes like this, which we confirmed in 2018, 2019 sampling also. You see the growth of this Picocyanobacteria in May, June, and then they consume nitrogen, phosphorus, and then they lay down somehow the red carpet for this filamentous bacteria to grow, Phenizomenon, for example, and then this even Dolichocrospimum also, this also filamentous bacteria. And then these are nitrogen fixtures. Some strains of Phenizomenon is the nitrogen fixtures. So they use nitrogen fixtures, and they have another important gene, which I will talk in my next few slides. Then this gives advantage to this particular group of cyanobacteria over this Picocyanobacteria. And then they consume all the nutrients. And then later in late summer or early fall, then these toxin-producing cyanobacteria, they dominate. They come and then they start producing. Just based on, this is the trend based on last 3 years, 3 to 4 years of sampling. This is from 2017. As I stated, we sampled instead of 4, we sampled 7 sites right here in 2017. And you see a lot of green. All months, every sampling showed green, which means cyanobacteria was present at all sites, spatially and temporally. And then, of course, Firmicutes, and then other proteobacteria. All this gray is autopet with proteobacteria present at the site. At genus level, again, this red one, Xenococcus, this was present. It's like more abundant than even look at this one, green one in July, like this vineyard buoy showed, and then this one, mouth of Provo Bay, and then Provo, you have higher abundance of Phenizomenon, but at other sites and at other sampling locations, Xenococcus was more abundant than any other cyanobacteria. And this is, again, Xenococcus is Picocyanobacteria. So in summary, these are like different what you call really operational taxonomic unit based on 97% sequence similarity. And then you see right here, this is measured OTUs. There were a lot. What I showed you in this graph, something whose relative abundance is greater than 1%. That means there were more cyanobacteria, there were more other bacteria whose relative abundance was less than 1%, but we did not include those in this analysis. So this is a comparison between short slide in 2016 and 2017. Total 20 species important were detected of cyanobacteria in 2016, but in 2017, total 36 species of cyanobacteria were detected, and more new species thrive, mostly toxic, and they were toxin producing, like mainly microcystis. But in 2017, we had more toxin producers detected through Ecogenomics framework. So these findings were published in Science of the Total Environment in 2019. If you are interested, please, you're welcome to access that paper. Again, the journal is Science of the Total Environment. This is from 2018 sampling, ecology and gene expression, I stated. And this is pretty similar graph, 7 sites. This is at phylum level. Again, green is cyanobacteria. And as you see, the relative abundance of cyanobacteria occurring, if I put 2016, 2017, 2018 together, 2016 has cyanobacteria, but some site has more, other sites, they had little less, right? But 2017 showed more prevalence of cyanobacteria across all sites. And now in 2018, you see the cyanobacteria present in all sample across all sites, across all sampling events also. So the relative abundance of cyanobacteria bloom is increasing over time. This is a very good trend to observe. But in terms of further resolution at genus level, right here you see this yellow. Again, this is Cyanobium right here. You see its relative abundance is higher than Phenizomenon, the blue one, dark blue one. and this Cyanobium actually is surprising. There's not much information available about this genus Cyanobium, but this is also Cyanobacteria. And based on some of the cell wall structures, the inside the cell wall, they are closely resemble to Xenococcus. So it could be just a misclassification, or it could be a different Cyanobacteria also in this analysis. But this is amazing to see how this Cyanobacteria, the small, they grow and they are thriving in Utah Lake. So, so this time, unlike 2016 and 2017, we focused on nitrogen and these two important metabolisms. And this is again through advanced bioinformatic tools. And what we found is at different sites. This is all combined, and then you see different sites right here. More reddish means higher abundance, multifold, and then this, if you go towards blue, you see right, less abundant or correlation. So at these on x-axis you see these different sites and then samples. For July, there's a multiple sample. For July, you see multiple right here because there are multiple sites sampled in July. Likewise, in May, there were multiple sites like that. So you see this relative abundance of these different genes. Most important, the takeaway message from this complicated graph is you see this nitrogen fixation. Nitrogen fixation is carried out by like several functional genes, and you see all these right here in this yellow area. They were all yellowish, and you will hardly see any blue or light blue. And nitrogen fixation genes were highly present in many of the samples. And likewise, right here, nitrate reductase, this gene is responsible, or at least suggests that there was significant capability of this water sample to conduct nitrate reduction. And again, this nitrate reduction is not carried out by cyanobacteria. As I presented my data earlier, there are other bacteria, that means non-cyanobacteria, are also present in situ. So what is happening when we look at from ecological point of view, when we look at the status, eutrophic status, and we try to understand what is going on in terms of nitrogen and phosphorus? Well, phytoplankton, algae, and more specifically cyanobacteria, they are consuming nitrogen and phosphorus. Their growth depends upon nitrogen and phosphorus, and their relative abundance also depends upon the concentration of nitrogen and phosphorus. But then you bring another variable, that means these other non-cyanobacteria present for other plankton, they compete with cyanobacteria for the available nitrogen. They make system limiting for nitrogen and phosphorus, especially nitrogen, because these cyanobacteria, these non-cyanobacteria, they convert nitrate to nitrogen gas. Cyanobacteria are fixing nitrogen to ammonia. These bacteria, they convert nitrate or nitrite to nitrogen gas. So your system is now competed by cyanobacteria and then these other plankton, and your system becomes nitrogen limited. So this is pretty interesting. The second graph is phosphorus. This is something which had been ignored over years, that okay, phosphorus is what we call a very conservative element, and its fate in ecosystem is primarily decided by physical and chemical factors, primarily chemical factors, because the gaseous form of phosphorus, which is phosphine, is very difficult to form. You wouldn't expect phosphine to form in freshwater ecosystem. So the only fate, but there are bacteria, they do contribute to phosphorus cycling in lake fresh environment. For example, a group of bacteria, polyphosphate accumulating organisms, which are primarily used or extensively used in municipal wastewater treatment plants to manage phosphorus, they are present in sediments. And under cyclic, anaerobic, and aerobic environment, they can cycle phosphorus in between sediment and the water column. So their contribution is ignored. And then there's other group of bacteria which have this phosphorus affinity group, phosphorus and transport, this phosphorus P starvation, P affinity group. And these bacteria, what they do is under phosphorus starvation condition, they can attract phosphorus, particularly absorb it and then store in their cell mass and then use it later on during phosphorus starvation period. So this data is pretty interesting. And as you see, it's pretty red right here. that means highly, higher abundance of these different genes across all sites. And you will see that's this P starvation, polyphosphate, and then phosphonate metabolism. These genes were present in high abundance, which tells that Ural lake is pretty dynamic in terms of these different bacteria. So my last slide is about these functional gene expressions. Again, these two slides shows that these genes are present. These genes are present. Again, I said presence doesn't mean that they're active, but in this slide, we specifically focus on 4 types of different gene: microcystin G, microcystin A. These genes, when expressed, tells that these bacteria, cyanobacteria, and these are very specific to cyanobacteria in general. They bacteria are generating cyanotoxins, and NIF genes is for nitrogen fixation, and PST gene is phosphorus affinity gene. And as you see, the dynamic, these concentration is copy per mil. These are expressed, that means active gene copy number. And right here, you see in July, they are expressed for example, Goshen Bay. And then right here, boom, comes July 1st, July 10th, 11th, the boom occurred in 2018, the boom occurred right in June, actually, towards the end of June. And then they're highly expressed right here, and then the expression continued. And you see microcystin G and A, that's where we see, as I stated in my initial hypothesis, here comes July, mid-July, and beginning of August, the microcystin, that means toxin producing cyanobacteria, they flourish. And this is quite supported with this functional gene analysis right here. Every site, mostly you see they're expressed. At some of the sites, they were expressed, but then highly expressed later on. And so at some site right here, they expressed highly after July end or so. And then most amazing thing is this NIF gene. See, at some site, it was constantly expressed. That means bacteria were fixing nitrogen. At other site, as we progressed in summer, May to June to July to August, this nitrogen fixation becomes more prominent right here. High temperature, nitrogen fixation, high temperature, increase. And likewise for phosphorus affinity also, with temperature, this the expression of this gene increase. But not only temperature, this is the need of bacteria when they need to use this, express this PST gene to scavenge phosphorus from their surroundings such that they can flourish. So conclusion from 2018 sampling, our data suggested long-term dominance of N-fixing cyanobacteria in eutrophic Utah Lake. The shift of cyanobacteria community was driven by both environmental factors and metabolisms dynamic, especially nitrogen and phosphorus in lake. Our data suggested that long-term dominance of phenizomenon and dolichospermum in the lake could have been attributed to the activation of nitrogen fixation and affinity genes under nutrient stress conditions. And cyanobacteria may function in early summer to initiate a starvation alert and store nutrients while utilizing the stored nutrients, or turn on and fixing during heavy algal blooms and nutrient limited conditions. Much of the data was generated through funding from USPA STAR funding mechanism. We enthusiastically acknowledge the support provided by Utah Division of Water Quality, and then stakeholder. And I truly appreciate, wastewater treatment managers that they've been great support always. I know if I need to get samples or go to Farmington Bay or something, I know they are there to help me and support my research. So they have been a great resource and help for me. All these excellent people out there in different municipal wastewater treatment plants. And then my last slide is just something different. We are doing this NSF, and my apologies, NSF funded citizen science project. Couldn't do much this summer, of course, because of obvious reason, coronavirus. But the citizen science, if you are interested in future in getting involved, we recruit citizens, we train them, and we provide them kits, the small kits, which can do chlorophyll, which can do nitrate, and then they report data to us. And then we also verify their data. So it's just not teaching citizen scientists about how to collect data. We have them do data collection themselves, and then we also verify their data by collecting water samples. So with that, I would like to thank you all for listening to this presentation, and I will be happy to answer any questions and any discussions. Thank you very much. Let me just, is it unsafe to fish in water that have active algal blooms? Yes, I would say, again, when we say active algal blooms, um, the question is whether this active algal blooms is harmful algal bloom, or does this contain any harmful bacteria. And as we saw, yes, most of the time, these heavy algal blooms, they contain bacteria. So I would say yes, if there is heavy algal bloom, it's unsafe to fish in water. Yes. So question 2 is, how can the process of eutrophication be reversed? Um, it could be reversed, of course, but this is not a short-term process. This is where we have to be patient. Sometime when people uh, tend to implement rules and regulations, we tend to think immediate effects of those new rules or technology and techniques. It takes time. Many lakes, for example, if I talk about exogenous nutrient input, that means nutrient coming to the lake environment from various sources, stormwater, agriculture, point sources, everywhere. And there are classical examples in China where they have done excellent job in cutting down the input of nutrient, and still the lakes, they kept experiencing this algae growth over next few years. And the reason is the internal recycling of nutrients. But if we minimize nutrient input, of course, this internal recycling of nutrient will also be exhausted over time. And then this reversal come, what we call a steeping point of ecosystem. This reversal process of eutrophication will start, and we start seeing good results. So the question 3 is it safe to swim in Utah Lake, or is there any risk due to organisms living in water? Well, I am not I'm not like organism guy, but in terms of cyanobacteria, again, it goes back to the first question: is it unsafe to fish? Much like if there are algal blooms and heavy growth of algae, just by looking at water, of course, qualitatively, uh, I think it's not advisable to swim in any water. But other than that, if the lake is clean and nice, then of course it's safe. It should be okay to swim in some portion of the lake, not everywhere, of course. Question number 4: are there other materials, chemicals, or organisms that can be introduced in the lake to neutralize or mitigate the damage to water? So, introduced in the lake to neutralize or mitigate the damage to water depends upon what damage we are talking about. Is it the damage because of nutrients, or is it the damage we are talking about any other chemicals? And so that the question is very subjective. But as I stated in question number 2, can eutrophication be reversed? Yeah, it could be reversed. And then in terms of introducing materials and chemicals or organisms, I think there is ongoing efforts in Utah Lake where they are adding some chemicals in some portion of the bay to mitigate algae. I don't have much details about the chemical process, but yeah, it could be done. But I would be really careful because again, one blessing could be curse for something else, right? And if we are trying to mitigate one contaminant, whether it's chemical contaminant or algae, the chemicals we add to any ecosystem could be harmful to other ecological processes also. I mean, one study we did, and a paper is under review. What we found is we look cyanotoxins from human health point of view, but cyanotoxins, what they do is when they are abundant, they're present in high concentration, eventually what happens, either they're degraded in water because of sunlight, or they settle down in the water column with sediments. Guess what? In the sediment water column interactions, there are various ecological processes. For example, carbon production, methanogenesis, methane oxidation, nitrification, which is ammonia oxidation to nitrite or nitrate. And these processes are carried out by bacteria. Now you have these cyanotoxins settled at the bottom, and all these bacterial processes are affected by the presence of cyanotoxins. That manuscript is under review, and we showed that yes, this is true. So we have to be careful. I hope this answers your question. So question 5, would aeration and bioaugmentation be useful method to reverse eutrophication in addition to decreasing nutrient loads? Aeration is good in terms of organic carbon. Aeration doesn't do much anything to nutrient other than enhancing nitrification. That means ammonia oxidation to nitrite or nitrate. And bioaugmentation, again, this is good for specific contaminants. As I stated, phosphorus is pretty recalcitrant, or what's a conserved element. You can't do much there in terms of phosphorus removal other than chemical precipitation. So our best bet for eutrophication is to control exogenous input of nutrients, whether it's from atmosphere or from stormwater discharges or agriculture runoff or any other sources. That's what we need to control. Question 6, could any of the conditions that exist in the lake afford an economic benefit if mined or harvested? That means fertilizers. One nice project I have in my mind I would like to do in future is harvesting phosphorus, as I stated, and the current paradigm understanding is which I kind of tend to believe also, because of just the mineralogy of the lake, the nature of the lake. The lake is highly alkaline in nature. That means its pH is pretty high, not only because of primary production, that means autotrophic oxygen production, but just by mineralogy, the lake is alkaline. Its pH is in the range of 9.5 at some locations, and always in the range of 8.5, between 8.5 to 9. So the phosphorus settles down in sediment, because phosphorus at high pH anyway tends to precipitate with the minerals. So one thing could be recover a bunch of phosphorus, because we will all run out of phosphorus in coming few decades. So recovering bunch of phosphorus from sediment could be a nice effort. Number 7, how does Utah Lake compare to other lakes in Utah in terms of pollution, like Bear Lake or Pineview Reservoir or any other major lake? So in terms of like Bear Lake, Pineview Reservoir, they're pretty clean, So I cannot tell about a percentage is 20%, 30%, 40%, but definitely Utah Lake is way more stressed out than these other lakes present. Okay, so looks like that was the last question, and I appreciate your attention and these excellent questions. Thank you very much.